How much happiness does chocolate actually contain? This is a visual representation of determination of serotonin and its precursors in chocolate samples by capillary liquid chromatography with mass spectrometry detection. The group members are Lecky, Matthew, and Samantha. Chocolate has been used for many years as an excuse for an unhealthy treat, but more recently it has started to become almost a superfood, offering antioxidant properties, compact source of energy, and now brain-altering chemicals that are synthesizing and being put into a bottle. But what does all this mean? With such a wide variety to choose from, is it all good for us? Or is there something in it that matters more than the rest of it? We no longer have to wait though to find this dark chocolate magic. Before we get started on the experiment though, what effect does serotonin have on your body? Approximately 90% of the total serotonin is located in the gut, where it's used to regulate intestinal movement. The remainder is synthesized in serotonic neurons in the central nervous system, where it has various functions with other chemicals. These include regulation of mood, appetite, and sleep, including memory, learning, and impulse. The purpose of this study was to develop a better way to determine the concentration of serotonin and its precursors, oxytrypan and L-trypan. By using capillary liquid chromatography coupled with mass spectrometry, they obtained their samples of 5 different varieties of chocolate with cocoa contents between 70 and 100% from a local supermarket in Madrid. Previous studies have been conducted on the same subject, but not been able to get the same resolution as what was found during this method. They include gas chromatography coupled with mass spectrometry, GCMS, and high-performance liquid chromatography, HPLC. The chocolate was crushed up into a fine powder and the fats removed and separated. Some of the initial experiments they ran exposed that to achieve a reasonable analyte peak separation, they would need a high aqueous content in the mobile phase. They tried multiple combinations of acetonitrile and ammonium formate, came up with the following conditions. The mobile phase was a combination of ammonium formate, 5 millimoles at pH 4 and acetonitrile. Initial percentage of acetonitrile was 3%, which held for 2 minutes, increasingly linear up to 7% in 1 minute, held for 5 minutes, and initial conditions in 2 minutes, continued on for 12 minutes. In general, the buffer concentration and the pH mainly affected the peak area, which decreased as these two factors increased, but only in the case of serotonin's peak width, buffer concentration had a significant positive effect, showing the highest values for this response at a higher concentration value. Regarding the focusing conditions in general, they affected the peak width of serotonin. The use of buffered injection solutions produced an increase in the response. Moreover, pH showed a strong effect on peak width in serotonin and L-trypan, while in the case of serotonin, the increase of pH produced a decrease in the peak width, and L-trypan, oxytrypan, this fact produced the opposite effect. The paper claimed that the main effect of pH on the peak width is probably related to the target pKa's, which is why acidic conditions were used. The injection plot, which is roughly shown here by SketchUp, the blue spheres represent the carrier solution to help the solution pass through, and yellow and white spheres are 2 separate ions with different surface areas. The mixed sample is injected into the closed loop and pushed through the column. Next, the column itself has a stationary face and small diameter shown here by the cones. As the sample flows through it, it has time to interact and hit both the walls and bind to the cones for long enough that the smaller particles can flow past with their smaller surface area. At the end of the column, the sample is spread out over the detector. It is also worth noting that it's not always 100%, and some of the sample may be left on the column, which is why in the study they let it run for over 12 minutes after the sample was extracted. The result was quantitatively found by mass spectrometry, which removes any excess solvent and the sample that does not match the mass of the ion to be detected. The ionization and breaking of the structures indicate the different shapes and compounds, and the quadrupole removes ions selectively to get the correct mass-to-charge ratios. Being organic structures, the rigidity of the analytes were not maintained with previous studies, but CLC enabled it to be far more accurate. The developed CLC-MS method, combined with the optimization sample extraction procedure, was found to be suitable to determine serotonin and its precursors at low micrograms per gram levels. Serotonin and L-trypan concentration range were detected, while oxytrypan was not actually found during this study. But what does this mean for you, the viewer? Samples of lower cocoa percentages and higher milk content had more L-trypan due to its milk, adding to the amount of the proteins present. However, serotonin synthesis requires vitamin B6, which is more also present in dark chocolate, meaning that somewhere along the line of production of milk chocolate, the vitamins may be lost.
Even with all this serotonin precursor goodness, make sure you don't substitute for things like meat, nuts, bananas, exercise, healthy diet, and when somebody offers you a piece of chocolate, your body will have no choice but to be happy.